Um, so one thing I would like to do in these is at the start of them is just ask everyone if there are any announcements. I think it'd be great if we shared any information that is going out to the world. We can talk about it in here briefly before we turn it over to our seminar. So any announcements for the good of the order? Yeah, Nathan.
If you're sitting real close to that door, there's 2 seats over here, and can I ask you to move over here so when someone shows up 5 minutes late, they're not sitting on the floor.
One announcement, so tomorrow morning at 9 a.m., donuts and coffee up on the 3rd floor little nook. If you're new to the building, good luck trying to find it. But just follow the scent of DJ's Donuts. If you haven't had DJ's, you want to be there for sure. So I'm going to turn it over to Tristan, if you want to introduce our seminar students.
Welcome everybody to today's seminar. I just want to briefly introduce Dr. Greer Dolby. I invited her as a speaker for the seminar because I think she has a really cool and integrated research program, which mainly focuses on how climatic and geologic aspects of the landscape can ultimately structure populations and affect adaptation and promote speciation. Just briefly, she did her undergraduate degree in Boston and did her PhD at UCLA. She then was a postdoc at Arizona State University for 3 years. And then she worked there briefly as a researcher. What was the official title? Research faculty. And then just this last year was hired as an assistant professor at the University of Alabama, Birmingham. So she's kind of split between 2 places at the moment. But I'll let her do the rest of the talking in terms of her research and the integration between geology and evolution.
Great, thanks. Hi everyone, can you hear me okay? Okay, I remembered to turn the mic on. Okay, so thank you so much for having me. I've had a really nice time chatting with people, and for those who I haven't gotten a chance to talk to, hopefully I'll have a chance to for the rest of the day or maybe tomorrow morning. But today I figured I would tell you a little bit about our work in the field of geogenomics, which might be a new term to you. And I thought, because it's probably a new term, define it early on. So really, I'm just today going to define this field as basically the deep integration between geological and genomic data. And I thought I'd start with a couple examples of how different changes in Earth's surface or climate can be expected to affect speciation or divergence, and then go into 2 examples. This one is all previous work that's published now, so if you're interested, there are papers I can point you to, and then go into some new data that we have on speciation of tortoises, particularly in the confluence of desert climate and river formation, and then finish up with kind of what we're working on now and how we're trying to tackle more complex landscapes and with new types of modeling. So I like to start with this nice little visualization of the tree of life, because to me it demonstrates kind of this really dynamic process of speciation, right, which we know is the process that has gone and formed all of the tips that we observe today, right? And so it kind of illustrates this bifurcating pattern, illustrates that dynamic process. What some of us spend less time thinking about is how, in many ways, that landscape that these species live on is equally as dynamic. And so that's one thing that I will try to convey to you today. And so 2 little examples here. Here we're looking at the North Pole about 18 to 20,000 years ago. And what you can see is the extent of glacial ice on land is much larger than it is today, right? And so the consequence of that is you had an extensive amount of Earth's global water budget that was stored in continental glaciers, and you can see the effect of sea level. So here we're looking at the Indo-Pacific, and you can see the major consequences that had for configuration of land mass and contiguity of the land. And here's what that looks like today. And if we move over deeper time, we might consider things like plate tectonics. in which case here I've starred the continent of India that's 65 million years ago was moving northward and would eventually slam into what we know as Asia and form what we know today as the Himalaya. So two brief examples to kind of show you that dynamicism. And how this might affect species is in two main ways. The first, and this is kind of this canonical or classic bicariant speciation model, where you have some change in the landscape from time 0 to time 1, be that drainage network formation or be that formation of some topography that isolates, here in my goofy cartoon, these little populations of beetles. Given enough time and given enough divergence, they might go on to form separate species. And so this might be a more kind of neutral example, and here's a speciation by adaptation example, where again you have this kind of homogeneous climate over some regional time spatial scale, and that climate maybe changes, an area becomes drier and an area becomes more wet. Populations adapt locally to those different conditions, and then given enough time, that local adaptation might prevent them eventually from being able to reproduce with one another and again form new species. And so both of these processes can give rise to lineage divergence or speciation. And we know that, I mean, that's well documented, but for me it raises a couple other questions, which is, one, are some types of geological or climatic processes more important for shaping these patterns than others? If so, why? And finally, can we actually measure these relationships and try to quantify these relationships in a little bit more detail? So the way we do this in my group is essentially going out in the wild and collecting sequencing genomes of plants and animals. I'll talk about a variety of different species today. And sequencing their whole genomes, although we also work sometimes with methylation and other data, and combining this, integrating this with collaborators who reconstruct the landscape or the climatic regime of that area over whatever time period of interest. And so this could be development of drainage networks, this could be looking at the sedimentological record, this could be, regional climate. And then one thing that I'm particularly interested in is how do we kind of develop these new quantitative or computational tools to allow us to integrate these data sets. And that's, I think probably most people would agree is like the real challenge with these types of projects. All right, so here's the first kind of case study or example I thought I'd go into. This is work from a while back at this point, but I like it as kind of an example of these types of relationships. And so here we go back to our visualization, again, looking at the North Pole, and here we have all this continental ice that's storing all this water, and as a consequence, what hopefully you can see over there is the effect that this had was majorly, massively changing sea level, right, from the LGM, from the last glaciation to present. So here's sea level today. Here's where sea level was about 20,000 years ago, and that's a difference of about 120 meters. And this was global, right? But that didn't just happen once. We've had these cyclical 100,000-year glaciation events of roughly equal magnitude going back about 1 million years. And so what does that mean? Well, if you're a critter that's an obligate coastal dweller, so maybe you live in an estuary, which I'll talk about, or you live along the coastline, here's where you're living during today, and here's where you're living when sea level is that much lower. So you can imagine the massive consistent movement of that coastline in this 3D model, going from present day sea level all the way to past sea level. And then you can imagine how this is going to be oscillating through time over the last roughly 1 million years. So we wanted to know, okay, given this kind of setting, what would that do to obligate coastal habitats? So things like estuaries, which is what we studied, which are these bodies, coastal bodies of water that tend to be a mix of marine and freshwater inputs. And so we set out to model where estuaries formed today and where they could have formed in the past. To do this, we developed a whole bunch of GIS or geographical information system models, and we used the geomorphic parameters of estuaries where they exist today, things like slope, sea level, a bunch of other parameters to estimate where they could have existed at roughly every 1,000 years from using a sea level curve from 20,000 years ago to today. And there are areas of the coastline where at low stand or when sea level was low, there was no habitat. So habitats were totally extirpated depending on the morphology of that coastline. Then there's some areas of the coastline where habitat was very fragmented. And from maybe population genetics, that very isolated habitats can have the effect, right, of leading to restricted gene flow and maybe divergence. And then you had some areas of the coastline where, and this is particularly wide coastal margins, where you consistently had habitat. And so that habitat was in existence as that sea level was fluctuating towards the ocean and then back towards the continent. So we developed these habitat models, and then we wanted to pair this with some genetic data. So we studied 3 species of these tidal estuarine fishes, so 2 species of goby and 1 species of killifish, and these are super small fish. And we sampled about 520 individuals across the 3 species, which we picked specifically because they co-occur generally in the same estuary, so there's this kind of biological replication. And this was back several years ago now, and so we used microsatellites. And we basically developed these models and did the sampling from roughly San Francisco, California, along the entire western US down through Nayarit, Mexico. So it's a pretty large sample area covered. And we wanted to figure out essentially what effect did this purported habitat redistribution have on the genetic relatedness and diversity and population structure that we see today. So we turned these GIS models into, to integrate these, we used an approach called approximate Bayesian computation, which I won't go into. But the idea was basically turn these into models where which populations were extirpated at which times, which ones were recolonized, and from what population. So we basically turned these models into these genetic predictive models, and we compared them against a whole suite of alternatives. So things dependent on ocean currents, things dependent on isolation by distance in different ways, or just some random recolonization models. And what we found was across the board for all 3 species, our sea level models were able to predict the number of lineages, the relatedness of lineages better than all of these alternatives. And so since that time, Josephine Stiller independently kind of followed up on this, and she worked on some population genomic work on the leafy sea dragon in Australia. And she kind of expanded on these results and showed that continental shelves that were wider, shown here in pink, in southern Australia, and this is consistent with our results, led to larger habitat availability when sea level was low, and that larger habitat availability led to higher observed heterozygosity or diversity observed today. And that's in contrast to areas where the continental shelf here and here is shown to be narrower. So this is consistent with what we had developed, but extended that further. And so we kind of developed this working model where we know from a variety of sources that tectonic and sediment processes are going to control, to a large extent, this shape of the continental shelf, the geomorphology. And what we're able to show, right, is the geomorphology is going to control where different types of habitat can form. And then when sea level changes against this geomorphology, depending on the shape of the coastline, you wind up redistributing these habitats, and that redistribution has a very clear genetic signature and explains well the patterns of genetic diversity and relatedness that we see today. Okay, so that's the end of the first part. What I like about that is it just kind of goes to show, I think, this integration you can do and how you can have a very physical landscape control habitat, the habitat control genetic patterns that we might be interested in. Okay. So I'll spend most of the time talking about this project, which is kind of data we're just starting to wrap up. And we'll move, from the coastline inland a little bit to orient you. So here's Arizona, here's Baja in Mexico, and here's California. And this is kind of a different set of features that we'll talk about, but what got me interested in it is there's this whole suite of species of different types of groups that are found to have species level divergence across this region. And a lot of it was thought to be due to the formation of the Colorado River in this kind of classical bicariant speciation model. And so we came out with this meta-analysis though, and we took genetic data, this is just mitochondrial data, from 33 animal, desert animal species. And we asked, okay, if you look at using FST, if you looked at the population structure, how much does the river explain population structure? And the answer was lower than we expected. So only about maybe 35% of this variation could be attributed to actually whether populations were structured by the river. And if we go to the geological literature, we know the formation of the Colorado River occurred roughly 5 million years ago. And this was a feature that people really expected and thought was structuring populations and communities in this region. And because this number wasn't higher, it led us to wonder about, is there pieces of puzzle maybe that we're missing? And so what we did was look at the several aspects of the climate across this area. And what we learned quickly was that here we have this really strong monsoon precipitation gradient. So this is dependent on, this is summer precipitation that I'm plotting here from brown going to blue. We're here in the east, so here in the Sonoran Desert, they have monsoon rains that come up from the south, from Mexico, come up and rain here, but that does not really occur west in the Mojave Desert. The Mojave Desert gets predominantly winter precipitation, and the Sonoran Desert gets both winter and summer. And so here I'm just plotting summer precipitation. You can see that kind of gradient. And you can also see the differences in vegetation. So here's the Sonoran Desert and here's the Mojave Desert. So this led us to wonder, okay, well, this gradient kind of co-occurs across the Colorado River, right? So maybe there's this climatic signal that we have overlooked. So to test this, we chose to work in tortoises specifically for a variety of reasons, but partly because they were thought to be this kind of canonical or this classic example of they specied roughly 5 million years ago in conjunction with formation of the Colorado River. And so we wanted to test, okay, do we see any signs of differential adaptation in the speciation history of these two lineages? So here again, I'm plotting monsoon precipitation, so summer precipitation from west to east. And this is a reduced sampling map, but basically on the left in black triangles are Mojave tortoises, pink circles are Sonoran tortoises, and what you will notice is there's a little hybrid zone right here in these yellow triangles. And so we developed reference genomes for both species, and we have sequenced almost 400 individuals at low coverage going across the entire range and down into Mexico, which I'm not showing. So you might wonder, for people who like speciation, what does that hybrid zone look like? This is what that looks like. So here's your little hybrid zone. Here's what those individuals look like on a PCA. Here are Mojave Desert tortoises. Here are Sonoran Desert tortoises. And what we seem to be seeing is that individuals from the parental lineages will make it into this zone, but they don't seem to backcross. They don't seem to come out of that zone and backcross and mate with the parental lineages. So that's just a little bit of a teaser. That's all I'm going to say about hybrids. So if you're curious, I'm happy to talk about that a little bit more, but that's what work that's kind of ongoing at the moment. So to get back to this kind of climate question, we approach this in two different ways. One is we subsampled that data set and we took 10 genomes from the core of the Mojave range, 10 genomes from the core of the Sonoran range, and we essentially asked, okay, if we take 1% of the genome that's most diverged, are there genes in this? And if there are, what do they do? And so for those who are interested, that was an FST cutoff of 0.63, which is pretty high. And then the mean pairwise FST is 0.34, and that's also relatively high. So these are definitely, species level, a species level divergence at this point. So we got 459 genes. It's a decent number of genes. We did a similar analysis here using those reference genomes, and specifically testing for genes under positive selection. So what are genes under adaptation, positive adaptation between these two lineages? And we got 421 genes. Now, I was excited because these numbers are like beautifully similar, right? No. So only 13 of these genes were actually shared between the two lists, which is probably not what you expected. It's not what I expected. And so we'll talk a little bit about why that is and what is going on. So I want to give a shout out. This is my graduate student Jade Meller's work that I'm presenting right now. And so this is going to the FST, so the population-based analysis, and asking, okay, where is that variation distributed? And it turns out about 75% of those highly diverged regions that had genes in them, that variation did not map to the gene. It mapped to flanking regions that were upstream or downstream. And so they probably have very little functional or putatively functional effect. And so we decided to get rid of that and focus on the genes that had putatively actual, divergence, either polymorphisms or insertions, deletions that were mapped to the gene itself based on the annotation. And this is what that looked like. So if you take this and spread it out, you can see there's a lot of changes in the promoter region of a gene that's going to control regulation. There's some change in the UTR and exon sequence, and then there's a lot of change in the introns, as you would expect. Because we're interested in putatively functional changes, we also ignored the introns, and the results for the next few slides I'm just going to show these putatively functional regions, so promoter, UTR, or exon. And so we took those genes, their putatively functional divergence, and we put them into a string interaction network. And basically this just asks, here's a set of genes, do they interact? Are they known based on evidence, experimental evidence? Do any of these genes interact? And so this is a statistically significant clustering, and what I've done is color-coded groups of genes that are involved in a similar function. And I'll just focus on a couple that are related to this hypothesis. And so the first one, what we find is this series, a subnetwork of genes where here these two genes are solute carrier proteins specifically involved in formation, regulation of energy across the blood-brain barrier. And after doing some literature review and diving into it, so both of these genes, one is involved in lipid transport across the blood-brain barrier, one is involved in glucose transport across the blood-brain barrier. And what's interesting about the blood-brain barrier is it plays an important role in an organism's perception of thirst and hydration state. So we think, in addition to maybe being an energy signal or energy homeostasis genes, that maybe these are also involved in maybe osmoregulation or water perception of water state, hydration state. And then the second two genes here are both mucin genes, which mucin 5B and mucin 5AC are mucin genes that are specifically expressed in the lungs and in the nasal epithelium. So when you get really snotty when you're sick, you can think mucins. And so these genes are thought to play a role in, well, one, when they're expressed and then they come into contact with water, they expand about 100-fold their volume, and so they sequester water. And we think maybe, so in addition to responding to pathogen threats, we think maybe they're also playing a role in sequestering water and preventing water loss across these kind of delicate membranes. And so in particular, 5B had a ton of exon changes that were non-synonymous, as well as a few gene duplications. Okay, so moving on, we have a set of genes involved in DNA repair and cell cycle checkpoints. The interesting thing about the cell cycle that will relate to the next slide is that this is a very time-dependent biological process, right? The checkpoints and the rate at which your cell undergoes cell replication is tied very specifically to temporal signals. And so CDC23 plays a specific role in this G2 mitosis checkpoint that basically checks, is there DNA damage before continuing with mitosis? So this is a bit of a complicated slide, but hopefully I can get through it. And so here, again, just another network. Two genes that we have are MED12 and MED26. This had a ton of changes, and they're both involved in the mediator complex, which plays a role in, plays a really key role in binding to the promoter and binding to RNA polymerase to begin transcription. What's interesting is it interacts with this gene that plays a very specific role in the hypothalamus in controlling circadian and circannual rhythms. So why does that matter? Well, that's another, again, kind of chronobiological process, like time-dependent process that relates similar to the cell cycle checkpoint of the previous slide. And there's evidence that these two things are kind of linked in an organism. Okay, does this answer our hypothesis or make any headway on it? Well, here's kind of a schematic of what's going on. So here we have these transcription genes that have a lot of divergence in these promoter areas that can control when a gene is turned on. And they interact with another transcription factor that specifically controls time-dependent processes like seasonal processes, like how birds figure out when to fly south for the winter, right? Well, it might make sense, right, that we find changes in regulatory regions, because if you think about trying to adapt to temporal processes, you don't necessarily need to change the protein structure, right? What you need to change is the timing of when that's expressed. And so regulatory changes are a way to do that. And we know that these two species vary. They have differences in timing of mating. They have differences in timing of when they lay eggs and the number of clutches. And they also vary in their activity timing. So for example, Sonoran tortoises will come out to bask in the middle of brumation in winter, whereas Mojave tortoises won't. So does this relate to monsoon precipitation? It's been proposed in the literature that actually all of these have diverged as a result of trying to match when rainfall is happening. And so what's interesting is a lot of species evolve seasonal behaviors in response to changing day length, right? Or differences in day length. But these are distributed essentially longitudinally. They have roughly the same day length. And so we think maybe it's rainfall seasonality that changes in time. And so these behaviors are changing in time. And so the timing of these regulatory genes that control these types of behaviors, that is what's driving that divergence. Okay, so just a quick finish for those who are interested in speciation and reinforcement. These were some nifty results that aren't super related to the hypothesis, but I couldn't resist. So this is my student Sarah's work, and we did the same interaction network. This is with genes under positive selection. And there's just two really cool results that I got excited about. The first is this really classic role of sperm egg recognition during speciation. So we found a whole bunch of genes related to meiosis and sperm formation, particularly the tail. But what's most interesting is here we have two genes that are under positive selection between the two lineages that are specifically related to formation of the acrosome, which is the tip of the head of the sperm, which holds these little enzymes that are what allows it to essentially penetrate and digest this jelly coat called the zona pellucida. So we also find another set of genes involved in the sperm egg recognition. And so this is predicted pretty classically by theory and by empirical evidence showing that as species diverge, right, the sperm and egg of one species need to be able to recognize one another differentially from sperm of the other species. So this is a pretty classic, consistent with very classic ideas about speciation. And here's the second one, which is a similar problem, but here we're looking at chromosome pairing and how that can go wrong in hybrids. And so we find a number of genes involved in the pairing and proper segregation of sister chromatids. And so in particular, formation of the synaptonemal complex, which all you need to know is this is an important structure for recombination to occur and to occur correctly. And so we found a number of genes. And so these are genes under positive selection that are playing a role in this kind of chromatid-chromatid assembly and segregation. And then finally, a set of spindle assembly checkpoint genes. And this has also been proposed to play a really important role in basically hybrid, not infertility, but hybrid dysfunction. Because what happens is here at the kinetic core, when the chromatids come together, there's a spindle at one end or at the other end, and there are microtubules that connect. And those microtubules need to reach a certain amount of tension in order for that cell to properly undergo cell division. And what happens is if these are mispaired, like for example, haploid cells or gametes coming together from different species, when these are mispaired, you don't have a strong enough cohesive force to actually form that tension. So this is again predicted. We're coming up with a bunch of genes that are predicted from the literature. Okay. So our hypothesis. Do we think that there's a climate signal? I think because of the regulatory differences in terms of timing of brumation maybe, as well as these mucin genes, I think that there's some evidence for a climatic signal in addition to a vicariant signal. And if there is, then why does it matter, right? It's easy to say like, well, whatever. That's not that important. And in and of itself, I think it's not that important. But I think it illustrates an important point, which is if we don't consider the multiple ways or several aspects of variation across the physical landscape, then if we limit our hypothesis space and we don't consider multiple potential factors, then we risk misinferring which one is important, right? We risk, if we didn't consider this, we would risk misinferring this vicariant scenario as maybe being the only one or the most important one. And so if we do that in a lot of different studies, I think we're potentially misinferring what are the factors that are important for shaping speciation and divergence. Okay, so where are we and where are we going? So I have a few slides on work that we currently have where we're in the fifth year of a 5-year NSF funded project to work on the Baja California peninsula with a bunch of geologists and biologists to try to make this model system for studying Earth-life interaction or geogenomics. And why did we pick this region? Well, there's for decades now been this really long-standing pattern of genetic divergence between northern populations and southern populations. It's not known why, but this is true. Like, pick your favorite critter, like lizards, snakes, rodents, plants, sunbirds, lots of insects. And it's been observed in over 80 studies. And so the question is, well, why? Like, what is going on? So we thought this would be a good area to study. And so we picked a suite of different species. We have a cactus, we have a desert bush, a couple of small rodents, and a couple lizards. And we set out to test a handful, 3 specific hypotheses. So the first is looking at, okay, this vicariance question, was there a physical barrier? And the long-standing one was, okay, this area, which is topographically low, it's like a valley through this region, is that maybe it was flooded at some point. And so the geologists went out and been doing field work for multiple years to reconstruct over like million to million year time scale, going back about 8 million years, what is the paleoenvironment of that region in particular? And that involved a whole lot of different data types. And we're doing whole genome sequencing to try to match codivergence, test for codivergence. And so I can tell you the super short 5-second answer, which is no evidence of a seaway. In particular, they actually find continuous non-marine sedimentation. Here, this is the mid-peninsular region. Here, this is Becky Dorsey's work looking at this stratigraphic record in this region. And this is Scott Bennett's photo. And here are a bunch of volcanoes that have since erupted, and they've been reconstructing faults and looking at the environment and been able to completely rule out that as a hypothesis. Okay, hypothesis 2. Maybe if we go back and we think about glaciations, maybe habitat has redistributed in a way that isolated populations, reconnected them. And so to test this, we're developing niche models to hindcast where habitat could have been. And then pairing this with whole genome sequence data, using those types of models and asking, okay, does habitat fragmentation based on these models explain present day genetic configuration? And this is my student Raul's work, and I can say, yes, this is still in contention. The northern populations and southern populations often were isolated during glaciations, and they responded totally differently. So this is just an example from Encylea farinosa, but most species follow this pattern. So we just got the whole genome sequence data for all of these species, but I don't have results on that yet. Okay, last but not least. So for niche, the same precipitation gradient exists that we just talked about with tortoises exists on the peninsula, where you have winter precipitation in the north, summer precipitation in the south. And so the question is, okay, maybe they're adapting to these different conditions, and that's more of an ecological signal. And so again, developing niche models to test, are these niches diverged? And then pairing that with whole genome sequence data to test, do we see signatures of adaptation? And then pairing that with RNA sequencing to ask, are reproductive physiological processes offset between the north and the south? And the answer to the first part is yes, the north and south have statistically significantly different niches for every species except the desert cactus, which makes them interesting and maybe intuitive sense. But you can see how different they are between the north and the south, with the south in orange and the north in blue. So what is the point, and why is this maybe a step forward in trying to conceptualize these problems? Well, we try to develop this project in a way where we were testing each hypothesis independently, because the truth is, there's many things potentially co-occurring, right? Climatic factor does not preclude a physical barrier and so forth. And so the idea that you have potentially more than one hypothesis being supported, and also the idea that species might not respond the same way. Some things can disperse better, some things might be more sensitive to climate. So the articulation and testing of these independently, I think, is forward progress. Okay, last. I have a couple slides just trying to show the kind of modeling that we're moving into for those who are interested, because I think I had some conversations about this earlier. But this is work from a master's student who just graduated and actually just started his PhD here in the medical program. But we were wondering, can we do a better job of actually, if we think about rivers, for example, can we do a better job of actually measuring and articulating different aspects of these potential physical barriers, right? And so we know if a river acts as a barrier is going to depend on the organism. Does it fly? Can it swim? Is it highly philopathic and doesn't want to move? But what about all the different aspects of the barrier itself? And so to do this, we use the contiguous US, and here are all the locations that we studied. We used published data. We basically calculated for a whole bunch of plants and rodents and lizards and snakes. We calculated FST across a river, and then we took river characteristics of that site. So we measured river width, discharge, which is flow rate, and the seasonality of that flow, and we detrended geographic distance as a signal. And we tested 10 graph structures and 38 total models. And I just am going to show you, this is the last result, the one result from the best fit model. And so here's our kind of mapping of that system. And in every single model we ran, we had this relationship, which is negative, which is not what we expected at all. And so this happened in every single thing that we ran. It was super, super consistent and confused us for a good long while, which basically says that here this path between river width and FST is negative. And that says the wider the river is, the easier it is to cross. And that is not intuitive, right? At all. Until you realize there's actually some biological support for that in the literature. It's actually really mixed. Sometimes yes, sometimes no. But when we started thinking about it more, here's two reasons why this potentially makes some sense. And if you look at the longitudinal profile of a river, in the uplands of a river, it tends to be narrower, and it tends to be higher elevation and deeper incision. So you have higher topography, and we were able to show this in the paper, but you have higher topography in these regions that itself is a barrier, right, to critters moving around the landscape. So then that's one explanation. A different explanation is in the lowlands, where you have like really alluvial dominated rivers. So here's the Mississippi. Then where rivers tend to be really wide, which is down here, they also tend to move a lot. And there's tons of evidence showing that these kinds of avulsions can move, essentially cut around and move populations from one side to another. So here you have facultative transport, right? And here you have isolation. So I only point this out because we would never have figured this out if we didn't take this kind of graph-based statistical approach called structural equation modeling. All right. What I'm hoping I've been able to show in some different examples today is Earth's surface is dynamic, but there's opportunities that we have to actually measure and quantify and test, do these changes actually shape speciation patterns and adaptation of critters? And with that, I would like to take questions. Thank you for joining today, and thank all the collaborators and funding sources. So thank you very much.
I was wondering, in your mind and sort of framework for thinking about these types of questions, how does geogenomics differ from what is, I think, traditionally called phylogenomics and phylogeography?
I think that's a good question. And I would say that the explicit integration of geological data and geologists is probably one point where it differs, and also more of a reliance on, so bringing in, primary data from more geological or climatic sciences, but then also bringing in maybe a little bit more from speciation genomics itself, right, as a bit more of a melting pot and bringing those two fields into contact. I'll say, I think it maybe answers a slightly different set of questions, because I think it really focuses on trying to model and understand what are the actual attributes about these physical processes that matter. And I think in my experience, a lot of times phylogeography is more about hypothesis testing, which is important, and then also maybe describing patterns. So I think I'd say maybe the deeper integration is probably where it differs the most, but also a real effort to focus on mechanism instead of pattern, and understand how these things are actually operating in nature. It's a good question.
Thank you so much for the talk. In the tortoise example, I was just wondering, I don't know if I did understand or not. So you use the river as like the main factor explaining the differences between like the population, but also you kind of like address that with the availability of water. And I don't know like how was like that decision to like going from like the availability of water explaining that like kind of like changes in the genomes explained by the river itself and not by the topography, and how is this story backed up by the geological record, or like what?
So that's a good question. So I think the idea behind that is that what we're seeing, so there's two things. There is, some structure related to the river, but what I didn't point out really well here is here we have a couple populations of Mojave Desert tortoises that you can see are on the other side of the river, right? And they seem to be perfectly described more by this 50 millimeter precipitation limit than they are by the river itself. And so, you can see they're mostly separated by the river. And this was like a different study that quantified the amount of divergence. It was for tortoises and a bunch of other species that calculated the amount of variance described by the river. But specifically the adaptation, those water-related genes that are what gave us the indication that maybe they are adapting to this difference in precipitation across that region. So it's kind of, we tested those independently in 2 different areas. We have the whole population data set, but we haven't like looked at isolation by distance and things like that yet, if that's what you're thinking.
Yeah, maybe. Or I don't know, like what kind of geological data are you like? Where's the geological data in your like?
Yeah. So
in this example.
right. So that's a great question. So I didn't include this, but in this paper where we looked at, so in this paper where we looked at distribution of species relative to the river, half of that paper is looking at the paleo river deposits in this region and mapping them and showing that what's interesting is in this particular area, which is exactly where you have those Mojave tortoises on the other side, is what we would call extraliminal, right? They're kind of on the wrong side. That's shared in 7 or 8 other species that are totally different. And it also coincides statistically significantly with where the Colorado River floodplain or paleo river deposits show this jut out where they aren't today, right? So it's the widest area of paleo river deposits, also coincides with where you have these extraliminal populations. And so yeah, that's a great question. I didn't include that. Probably because there's already too much in the talk, but yeah, so those in this paper, if you're interested, that's what we used to show that. That's a good question. Yeah.
Are you ever, with any of your studies, able to infer rates of landscape change versus rates of genetic change?
We can definitely estimate rates of genetic change given some caveats. We have not estimated any landscape change rates. I would not personally attempt to do that. I would kick that off to a collaborator who could probably do it better. Yeah, but that is, I think of absolute times, right? And so if you do phylogenetic tree reconstruction, you estimate something it diverged maybe 5 million years ago, then for me, I kind of just hit the literature and look at, okay, what has changed on that, given that time frame? What do I have to worry about? What do I not have to worry about? Yeah, Jack it in.
That was really fun. I was curious in the last vignette that you presented. So maybe building off both the Catherine's question and the previous question. FST, we talked about this a little bit today, is like shaped by processes acting on the entire history of the set of samples that you're interested in. And thinking about the map of the drainage you showed, it seems like lots of other stuff has happened, like a lot of that area is glaciated and the rivers have probably changed in size. I was curious if you could get at some of the timing of the processes that are driving divergence by looking at like sharing of Greer alleles or like LD information to learn if there is no dispersal in the now versus no dispersal across rivers ever versus dispersal historically versus now, trying to get at the timing of the
I love that idea, and I haven't. I would like to do that. I think because we basically just grouped populations as being on one side of the river or the other today, then I think the idea of it's, in my mind, averaging over the history of however long those individuals or their ancestors have been isolated by the river. So I think of it as maybe you're adding noise to the signal, or it's an averaging over a time period. But you could do that in particular using, if you were to sample the genome and partition it into maybe rare alleles or like whatever the longer term signal is. What's cool about this is you can use the same exact structure, you can run through 2 different data sets, and you can figure out what the relative controls are, right? What the relative values are, and say, okay, there's a significant decrease, for example, or historically the value is much larger than when we look at more recent. So I think that would be a great area to work in, movement forward. It's one of the benefits of using a generalized structure like that,
So with your geogenomics group, sort of Baja, you've got, I guess, a lot of geologists as well as biologists. So what did the geologists get out of it?
Time with us. No, I'm kidding.
The reason I ask is because there was a geogenomics group with the geologists, and in that case, the geologists really wanted to address geological questions using biological data. Turned out to be harder than they thought, but is that the case here? Are there geological questions that your colleagues want to address?
No, not explicitly. So we picked people who've historically worked in Baja, right? So they knew the area. They really just wanted an excuse to go back and work more. And so that worked out for us. I mean, they definitely got a good deal of funding and funded a bunch of, master's and PhD students and had an RU down there. What I have found in the going kind of back and forth is I think they appreciate having their knowledge have a bigger impact than just in their knowledge domain, right? Like extending that knowledge and informing a completely other field that we would never be able to do without them. So maybe increase importance, whole bunch of, papers, we cite all of their stuff for sure. And I think some of them are just curious, right, about this. But I do think that that's an issue. Is your intent to fundamentally integrate the fields, or is your intent to use one to inform the other? And that's completely fine. But I think being explicit on both sides about what the intention is. But then also, it has, how science works, right? It has funded them to be able to go off and do a whole bunch of other things that they would never have done. And there's been a lot of discovery there because, they go into a place or studying something because we need an answer and they never would have done that otherwise. And they find new things, right? It's a different motivation. So you kind of wind up sometimes coming up with totally new lines of inquiry or findings than you would have otherwise had.
You have talked about living species.
Yeah.
That's what you've been working with. And I just wonder what paleontology could contribute to some of these hypotheses that you've been dealing with. For example, in Baja California, this is remote from my field. I understand that some subspecies of the redwoods were forced by the Pleistocene glaciations to migrate into Baja California, and this has been discovered by fossils.
Yeah.
And if those fossils only extended down, say, part way through Baja, then possibly this could account for environmental changes that cause the distinctions between the southern and northern biota.
Yeah, that's a good point. So we do have midden data that goes back kind of this intermediate time scale, right, through the last glaciation in Baja that is, kind of like pack rat midden data where it'll preserve some of the plants that were living at the time. And so some of that has been useful in trying to reconstruct that evolutionary or that ecological history in the region. But to your broader point, I think that there's a lot of potential to integrate the two. I haven't spent a lot of time thinking about how to do that. Within macroevolution, that's done way more commonly, right? When you're looking at speciation patterns and when you're working with phylogenetic trees, you can easily work in the molecular data and the fossil data. But I think there's a lot of avenues where paleontology could contribute and resolve questions and maybe even bring in new hypotheses. I just haven't. It's a great question. I haven't gotten there yet. Yeah, I'm open to ideas and going in that direction. Anything else?
I'm just curious, in the hybrid zone between the two tortoise Have, I guess, in the living individual hybrids, have you found that it's like their genotypes tend to be deterministic, where like there's certain genotypes that just you don't see alive in the wild?
Yeah, that's a great question. Jade's work, so I just showed a very small portion of that whole data set. The rest of her PhD is essentially sorting through that larger data set. So we haven't actually looked at the genotypes of the hybrids themselves yet. That's like, as soon as she gets out this paper, that's the next step. But what I'm also interested in, not only specific genotypes, right, but if you look at adaptation, or if you look at the genotypes of the genes that we think are under purifying selection, right, or sorry, under positive selection, how do those shake out across the hybrid zone? I think that would be really cool. Yeah, and I think, yeah, so I think that's on my list. I'm very excited for that.
Yeah, just following up with that question. I think you are kind of betting for adaptation, but I think the genes that are probably more responsible for adaptive isolation are those that are popping up. So just, I think when you start looking at your hybrids and individuals, like doing analysis of the progression, are looking particularly at genes that are associated, might be associated with reproductive isolation.
Yeah, I totally agree with that. That's a great point. Yeah.
So great talk. I was interested in your network analysis and the string.
Oh yeah.
And I think you mentioned once that there was, or you mentioned the gene duplication.
Yes.
Have you run into any issues where there's more, like you found duplications in course or something that may not be in the database or references, and have you had to work around that at all?
Thankfully, not yet. But you know, we've come up with studying tortoises. We've come up with a handful of gene expansion events. Mucin so far, like the biggest one. I didn't really go into it, but we haven't seen duplications of unknown genes yet. They even might be there, but they haven't come up partly because they're not annotated, right? And so we don't focus on them that much because we have no idea what they do. There's always that issue when you're translating your genes to what's known in gene ontology databases, right? Because you're just limited to one, what's in the database, and that's all based on model species, but also limited to like you assume the function is the same. And when you see gene family evolution, you have like a lot of duplicated genes that have different protein domains. You know that that's probably not true, right? So it's a problem, and I'm glad that I haven't had to deal with it yet. And if you have to deal with it, then I have a lot of empathy because I don't know what I would do with that. Is that it? Cool. Thank you guys.